Welcome to the video on significant change in assay on stability. So you might have heard about the terminology as stability, assay and significant change. So whenever there is change in the assay or potency beyond the specified limits, it will come under the category of significant change. So assay or potency is required to maintain the quality, purity and strength or label claim of the formulation, and also it is required for the drug substances. Generally, the drug product assay limits are 95% to 105% of the label claim or 90 to 110% of the label claim. So, whenever you consider the drug product for European region, the assay limits are 95% to 105% as a general rule or as general guideline, and generally for US it is 90 to 110%. For drug substance it is more stringent and might be from 98% to 102% or other limits as given up by the EMF holder or the manufacturer of the drug substance. So what is significant change or what is out of the limit assay values? Significant change for assay results as a change that is greater than 5% of the labeled amount of the API. So if the initial assay is, for example, 100% at the initial, and if it decreased by more than 5%, then it will go into the category of out of specification and it will be a significant change. So greater than 5% change from the initial amount or from the label claim as given by the applicant, it will come in the significant change. So the assay value may be lower or higher, or there may be variation in the assay values. So why there is change in the assay value on stability? See, the stability involves exposure of the drug product to the moisture, temperature and light. So there may be degradation which leads to lower assay and increase in the impurities. API may break down due to storage conditions, maybe due to the temperature or humidity or combination of the both, or storage for the longer time. It may result into loss of assay or potency. It may be due to instability of the formulation. It may also be due to failure of the packaging system or due to the pack integrity. Also, the instability of the API may be due to the interactions with the excipients or interaction with the environmental conditions, and also may be due to the degradation. So these are the reasons. Now we see what happens due to significant change in assay. So less potency will have the less efficacy or less action. Increase in impurity will lead to less safety. So safety concern if assay is low or assay lower side due to increase in the impurities. Reformulation as a failure in stability. So the product will be required to be reformulated because of the assay failure. Assay failure may also lead to less shelf life time period. And if you have applied the product or you have filed the product and you come to know that there is a significant change in assay, that time the regulatory approval may take longer time. Now we will see what are the best practices to avoid the assay failure. So first, Best practice is in formulation R&D for selecting the composition, selecting the manufacturing process and packaging system. Then understand the intrinsic nature of the API and intrinsic nature of the formulation. Then understand the storage condition impact onto the API stability and onto the formulation stability. Whenever there is change in accelerated stage, go to intermediate stage and see whether there is similar impact or not, and then go to long term stability condition to study whether there is significant change in assay or not. So if it is only at the accelerated condition, you can have the data of intermediate condition. So this is regarding the significant change in assay. I hope you might have got good idea about the topic. Thank you for watching the video.